You can start better.
It's 75576 already. So hi, I'm Anwesha Naskar. I'm fifth year integrated MSc students. I hope all of you know me, like some of you know me, maybe. And I was doing dissertation in Akash Golyani's lab in STEM, MCBS. And so as you can see from the title, my primary study was on Fyn Kinase. So what Fyn Kinase is, how do we study it, like what are the tools we use to study Fyn Kinase. I will talk about all of them, and mostly it will be about the biosensor rather than the Fyn itself, because like all of my project was on developing a biosensor which can detect Fyn Kinase inside a cell, in a live cell, obviously. So we briefly start with the introduction of Fyn. I don't know, but this is not shifting, right? Huh. So I shall start with the brief introduction of what Fyn Kinase is. So as you can see, it's a tyrosine kinase. It's a kind of tyrosine kinase which is embedded in the lipid membrane. So in the left, in picture A, you can see that there is active Fyn, which is totally unfolded, and it has 4 components to it, like SH1, SH2, SH3, and SH4. And it's absolutely sitting there and doing nothing. Then next to it is a PAG. What is PAG? That doesn't matter right now, but it's just to show that the Fyn is active, but it's not interacting with the PAG protein. As we move on, so here in the picture you can see that Fyn has mediated the phosphorylation of PAG. So as soon as the Fyn phosphorylates the PAG, the tyrosine 317, which is situated at PAG, get phosphorylated, and then it connects to CSK, and then further down regulation of a cycle continues. So this picture is basically to show you how active Fyn and an inactive Fyn looks, and why are we like interested in knowing what an active or an inactive Fyn looks like. That you'll like get to know in subsequent slides. So then next to it, it's in picture A, there is inactive Fyn. As you can see, all of them are like folded together, properly binding to each other. And then there is this tyrosine at 531 position, which is phosphorylated. So this phosphorylation at position 531 is making it inactive. So the kinase right now cannot do anything. It cannot interact with any other kinase inside the cell or PAG or whatever. So a brief history of kinase, or like a brief schematic diagram from where it origins. So Fyn kinase is a SRC family of kinases. So there are certain kinases, like non-tyrosine type of kinases, which are grouped in one family because they all behave up to like similarly, or their composition, as you can see in the Fyn kinase structure, most of them has similar structure. They all have SH1, SH2, SH3, and SH4 components to their kinase structure. So as you can see that in the picture on the right hand side of the screen, there is this expression pattern of SRC, yes, FRG, ERK, Fyn, Lin, HCK, LCK, BLK, and FRK. So all of these kinases are SRC family kinases. Okay, so as you can see that there are 4 kinases, namely SRC, yes, ERK, and Fyn. These 4 are ubiquitously present. and what do we mean by ubiquitously present, is they are present everywhere, like all of the cells in your body. So like, what's the big deal? You say, you can say that, like, what's the big deal? Why is it like, why do we need to know that it's ubiquitously present? If at all, like, why is it present there? Why do we need to know about its existence? So as you can see that other hello. Hello.
Hello.
Can everybody mute their, like,
Everybody.
You're not asking questions.
mic?
Can you kindly mute your microphones? You're not asking questions.
Yeah, like I can open the floor for questions. Then you can ask. Hello.
Yes, you can continue.
Okay. So as we can see that SRC, yes, ISERC and Fyn are ubiquitously present. That means they are like more or less present in every cell in our body. And the other kinases such as FRG, that is present in myeloid cells, brain cells, and you can read that what rest of the kinases are present in. So our study particularly focuses on Fyn. So the question is why? Why are we focusing on Fyn particularly when there are like so many SRC family kinases? They are like present in so many places. So the answer to that is Fyn, which is ubiquitously present in all of the cells in our body, are known to regulate cell migration. Listen to this carefully. Cell migration, epithelial to mesenchymal cell transition, cancer cell metastasis, and immune response. And when we look at the like neurogenic properties of this kinase, a lot of paper have shown that Fyn kinase is particularly a key nodal kinase to axonal guidance, patterning, and synaptic functions. So yeah, it's pretty much important. So as we move on, that but you have to have a particular study which you want to look at. Like you want to correlate the Fyn kinase to a particular study you want to look at. So what our lab was doing, that they were concentrating on the cellular migration aspect of the Fyn kinase. So as we move on, so all of you must be studying about different types of kinases, proteins, and I don't know, enzymes maybe. So many of you might know a lot of techniques about detecting kinases, proteins, and enzymes. So can you all quickly think of some detection techniques to at least I don't know, maybe detection of kinase, of protein or enzyme, like technique which is related to the detection of any of the kinases or protein. Can you like think about it for a minute? Or like anybody wants to answer, they can answer.
Anwesha, I have a question. Can I ask now or at the end I have to ask?
Yeah, you ask. It's okay.
Where are these Fyn kinase where are they mostly present? This Fyn kinase.
Did you hear the first slide?
Yeah, I heard, but I think I missed it or I have some doubt.
Yeah.
I didn't get it
So
properly. Where are they from?
yeah, so Fyn kinase, as you can see, that Fyn kinase is present in it's the expression pattern of Fyn is ubiquitous. It means it's present all over your body, like everywhere in your body, every cell. So, but there are specific function of Fyn that I discussed. I don't know whether you heard or not. So we are targeting to study one particular function of Fyn, and that is cellular migration. So we move on with the question that how to detect Fyn kinase in a cell. It's very like specific question. My question was how to detect protein kinase or any enzyme inside a cell.
One question though, in cells where it is found, like you
No,
mentioned,
no,
it's
no.
found.
No,
Which
no.
part?
My question is how to detect any of the kinase, protein, or enzyme.
I was just
Like
following
anybody.
up Sumu's question. I was just following up Sumu's question that he mentioned where it's found. You mentioned in cells. So where is it found in cells? Which part of
Yeah,
cells?
yeah. They are mostly attached to the lipid membrane of the cells. So actually, there is this study which says ki, so as I said that we are studying cellular migration. So there is this study which shows that ECM is extracellular matrix, which has integrin, fibronectin, or like whatever, like matrix kinases, and it interacts with the so the active Fyn. So there is this mechanism called FAC mechanism. That is a very big mechanism. Like it's not possible to discuss that big mechanism here, but there is this mechanism called FAC, which in turn interacts with integrin. So if you picture a lipid membrane here, in the picture you can see the picture A, right? So there is this lipid membrane which shows that it's attached to the lipid membrane, but there is this picture of FAC which is not shown. So what FAC does, it interacts with integrin. Integrin is present outside of the lipid membrane or your plasma membrane, which in turn interacts with ECM. So it's like the ECM sends you signal that you can move ahead, or here you can get a lot of matrix to move ahead. So ECM signals through the integrin to the FAC, and then in turn it signals the Fyn kinase to further like act inside the chemical biochemical pathway. Hello?
Yeah, okay. So these parts, SH4, SH3, SH2, SH1, these are all parts of Fyn.
Sorry again.
So SH4, SH3, SH2,
Yeah,
SH1,
yeah.
the tyrosine.
Yeah, I told in the introduction only these. So so I'll repeat again. SH1, SH2, SH3, and SH4. This kind of, so this module is inherent to SRC family of kinases. So all the SRC family of kinases you want to look at, they look almost like more or less similar to this structure. So this picture was just to show that what inactive and active Fyn looks like. So is that okay? Hello? Hello?
Oh, okay.
Yeah, so I move on to the next slide. So I asked you the question ki how to detect any protein kinase or enzyme. So there are various studies to actually detect it. So one of you might say you can detect it from ELISA. One of you might say that it can be detected from, I don't know, immunofluorescence. You can detect, you can pull out the whole kinase from the cell and then detect it. So why are we like so interested when there are already ways to detect a kinase? So why are we taking headache to like make a biosensor out of it, or like what is the big deal in searching Fyn kinase, and like why do we need to study Fyn? So these are the questions which will be answered in subsequent slides as I move on. So here is a toolkit for making a biosensor. So first of all, we start with the fluorescent protein. what are fluorescent protein? So the first fluorescent protein, which was isolated from Aquaria Victoria. So you can go to the source fpbase.org. It's linked down. So it's fpbase.org protein at AVGFP. So the first protein which was found from this jellyfish, Aquaria Victoria, was named AVGFP. So this AVGFP was isolated, and then some guy called Carrot crystallized the structure and found out that your green fluorescent protein particularly has a beta barrel structure, and inside this beta barrel structure is the chromophore. So you can like see the beta barrel structure, and to the right of it is the chromophore, which is fluorescent, which is the fluorescent part of the green fluorescent protein. And so the beta barrel contains this chromophore inside it. So this was all about the basic structure of green fluorescent protein. So as we went ahead, there were many marine animals and marine microorganisms which were detected, and then a range of fluorescent proteins were made out of them. So GFP gave rise to a lot of fluorescent proteins such as EBFP, ECFP, EGFP, YFP. And there is this molecule from I guess diatoms, I don't remember exactly, but it was called DS red. So DS red gave rise to all the red shift part of the spectra. So you can see on the right side that TD tomato, M tangerine, strawberry, cherry, all the red and pinkish fluorescent proteins, which was that was derived basically from DS red. And yeah, so the source was a jellyfish, which was Aquaria Victoria, and then they discovered fluorescent protein. In 2008, Atushi Miyawaki, Roger Y. Shen, and Michael Chalfie got Nobel Prize for their discoveries. Actually, Miyawaki pioneered these studies of isolating these fluorescent proteins, and then Chalfie and Roger Shen did many things to it. So there are as in 1956, when Atushi Miyawaki actually discovered the fluorescent protein, he didn't realize that it can like what all it can do. So later on, after 1956, research went on. He actually didn't do much with the green fluorescent protein himself, but then some other scientists like Roger Shen and Michael Chalfie, they with their groups, obviously they did a lot of experiments. So here is a quick video of what green fluorescent protein can do. So as you can see, these are bunch of cells which are green right now. Okay, so I'll tell you what these colors signify. So the green color signifies that it's in mitotic phase. So as soon as it changes color and go to red, it means it's in interphase. Okay, so what we do is we genetically modify the DNA material, and we put a fluorescent protein tag to it, and then we transfect the cells with these fluorescent proteins to further watch what these can do. So here is a quick video how useful fluorescent proteins can be. I hope all of you saw this. Hello, I need response. Hello.
Yes.
Yeah, so the green cells which you saw, okay, I'll replay it again. So the green cells which you saw, which quickly turned into some red, then again green, then red. So the green cells were actively in mitosis phase, and the red cells were in interphase. So these were like small things you can do with genetic engineering and tagging fluorescent protein to your genetic material to see various stages. So this was very preliminary experiment to fluorescent protein, and nobody actually thought that what else you can do, and it took a long time to actually realize that it can be made into a physical biosensor, a molecular sensor which can then go inside a cell and detect a particular protein and detect its movement. So there is one example from Miyawaki's paper in Cell. So here is a mouse. So on day 1, so they were studying benign and malignant tumors with the help of this newly found fluorescent protein technique, and this technique was called Fushi. So this Fushi technique was expressed in HeLa cells, and this was the video which you saw. So yeah, cool video. What else? This is not like, this is biology happening, but what new you can do with it? So then Miyawaki and Shoukawa Soano went on, and then they saw benign tumors and malignant tumors inside the cell. Actually, they just injected those mass of cancer which were tagged with green fluorescent protein inside the mouse. So this was on the left side, on the right side. A was day 1, where it shows, the first panel shows the place where the benign tumor or the tumor mass was injected. Then subsequently they imaged after few hours on the same day that it showed yellow fluorescence. So what yellow fluorescence meant that the cells were actively in mitotic phase, that they were growing. And yeah, obviously if you put cells in an environment which is favorable for its growth, it will definitely grow. So on day 1 they imaged it, and they saw that so this isn't very good spatial resolution to a picturing so for imaging, but still it wasn't invasive. They saw it under laser light, and then they saw that it's actively mutating on it's actively on mitosis on day 1. On day 16 they saw all of the cells were in interphase. They are like not dividing anymore. So they monitored it for further days, but it was in this state only. So that's how they concluded that the tumor was benign. And further in the image D, you saw you see a mass of cell which was being injected to the mouse on day 1, and then on the same day they imaged it. Yeah, it was perfectly dividing. It was in mitosis phase. So when you take green images and you take red images, you overlap them, then you get this sort of bluish red kind of a thing. So this is marker that those cells are still actively dividing. They are in mitosis phase. So on 16th day, you can see that cell mass has grown bigger. So as you can compare them side by side, that in C, which was benign tumor, like which was found to be a benign tumor, the cell mass wasn't bigger, or like it was there, it wasn't doing anything, it was an interface. But on 16th day, the malignant tumor mass, which was spreading and which was still in mitotic phase. So yeah, you can say that this tumor mass is like growing further. It's not benign. It's not staying there. So this was one use of this Fushi technique. So after the fluorescence protein was invented, they did this minor things. They introduced Fushi, then they tried some preliminary experiments. And not until 2001 and 2011, sorry, this started again. So, okay, so not until 2000 and 2000 to 2011, people realized that this can be made into made into various kind of sensors. So this paper was Guliani et al. This was my PI's paper. It was published in 2001, which shows some monobody libraries, which is nothing but some proteins. Okay, so these merobody biosensors were tagged with a dye. So to visualize anything, you need to. So okay, so when you do visualization or radioactive studies, basically what you do is you tag radioactive labels, dyes to a molecule or a compound you want to see. So similarly, this approach was used that they made a sensor, a very basic sensor, 3-component sensor, which had a dye constituted to it, a variable region which can attach to SH3 domain of the Fyn kinase. We will further see. So in figure A, you see this merobody biosensor, which has a fluorescent protein, a variable region, a cylindrical variable region, and a dye. So in B, you see this biosensor in action. So what happens, as I showed you in the first slide, that when Fyn is in inactive state, it's totally coiled up and like it's sitting there, it's doing nothing. So this is Fyn in inactive state. Left hand side, the Fyn is in inactive state and it's not interacting with the dye at all. So then we see that when the kinase need to be active, it sort of opens up and then it sticks to the biosensor and bam, it glows. This was the next kind of sensor was made. It was made in 2011. So till then, people were just putting fluorescent biosensor in gene you want to tag and see it inside the cells. So actually the question arises here that when you can tag DNA or tag a protein with GFP and visualize it under the microscope, what was the need to make this sensor? Like, why to waste 10 years making another sensor when you can already tag the fluorescent protein and see, like, pretty much see what happens inside the cell or inside the nucleus or how cell is behaving. So the answer to that is a GFP protein can be tagged to the fluorescent protein, but then you can see fluorescent protein in all kind of cell, all over the cell. You do not see the active phase of the kinase or the inactive phase of the kinase. So here, what the sensor does is it's not detecting when the Fyn is inactive. Means, so there is a place inside the cell where this Fyn is sitting, just sitting, it's doing nothing. Why would the sensor go undetected? Like, obviously, if you tag a fluorescent biosensor, it will be expressed with the Fyn sensor and it will glow and detect it. But this sensor doesn't do that. It only sticks with the active part of the Fyn kinase, which is SH3 domain of the Fyn kinase. So as we move on, people did a lot of studies with this similar technique. A lot of people got different ideas. Then they saw a phenomena called FRET happening, a lot of substrate to biosensor interaction happening, and they just went experimental, and then various kind of biosensors were made. So these were the notable mentions because our study, like our biosensor, were based on these particular studies. So I'll talk about these only. Like there are various other biosensors you can check out, but these were the like key mentions which helped in further study inside the lab. So on the right, there is this phenomena called fret happening. Okay, so just to clear out, molecular biosensors are like divided into two categories. One is substrate based biosensor, one is conformation based biosensor. So like these are the broad classification of biosensors. So right now what we are looking at, a substrate based biosensor on the right. So you can see there is a SH2 domain. So this is like genetically modified. You are just taking SH2 domain of the kinase. You're attaching an ECFP fluorescent molecule to it. Then there is a linker which has a Fyn substrate and which is then linked to YPET. So this is all like happening happening at molecular level. You have to design it. So as you can see, this Fyn substrate has a Y component, which you will see that further, which gets phosphorylated for the biosensor to get active. So here, what is happening on the left side? The substrate Y is not phosphorylated; it's dephosphorylated. So in the left, you can see that the SH2 domain. So SH2 domain was there to get phosphorylated, and which makes it further like inactive. You can see on the right side that when you phosphorylate the SH2 domain, it sort of get inactive. You can still see some spectrum, but it's not fret. So let's explain what fret is here. So you take two fluorescent proteins. In this case, it's ECFP and YPET, and you need to get them close together. And by close together, I mean to the affinity, like to the vicinity of 60 angstrom. And if they're far apart, like if they're anything more than 60 angstrom, they won't interact with each other and they won't show fret basically. So what's happening here, that in the active state, when you are like sort of exciting ECFP at 427 nanometer, it emits a light which in turn activates the YPET, and it sort of shows the emission at 256 nanometer. So yeah, this was pretty much it. That this was the basic fret sensor that was made. It has a very hodgepodge kind of what you can say design, but yeah, this was the preliminary thing, and rest of the biosensors were obviously made on this. So on the right, you can see that the SH2 substrate, when it get phosphorylated, and even if you use a laser to sort of activate it at 427 nanometer, it shows an emission at 478 nanometer, not the 526 nanometer, which was the emission rate for YPET. So it's happening because YPET is far apart from ECFP to interact with it, like way more than 60 angstrom. So for the basic need for fret to happen is that both the fluorescent protein stays within 60 angstrom distance. So basically you can just see the emission spectra for 478 nanometer in this case. So yeah, these were substrate-based biosensor, which sort of pioneered for the next few designs. So this was the Fred biosensor made in the lab by Mukherjee and Singh. So the previous model was an inspiration for this model. So A shows the very crude picture of what this biosensor does. So this kinase is in off state. As I showed you that Fyn is in closed state is off. So here the kinase is in off state. The sensor comes and doesn't interact with it. Okay, this was the basic concept of making the sensor. And, in the next, like in downwards, when the kinase is on, the kinase is on as in the SH3 domain of the kinase is exposed. It binds to the sensor and the sensor glows. So this was the very basic idea of it. So they went on to make the whole biosensor. Okay, so the whole biosensor, so you can see the kinase in orange, and it obviously has components SH2, SH3. So the kinase was cut down and was optimized to SH2 and SH3 because people saw in previous studies, people saw that SH2 and SH3 and the this kinase orange part is basically important for the Fyn activity, like for the Fyn to activate and show activity. So what it does, like you can focus on the image B. So when the kinase is in inactive form, kinase is bound to SH2 and SH3, and the SH3 is tagged with M-cerulin. Okay, so in closed form, it shouldn't show Fred, right? So when you illuminate it at 437 nanometer, the M-cerulin emission spectra is at 475 nanometer. So when it's in closed state, it will show you emission at 475 nanometer, and you know that the kinase is not active here. So when you go to a place where you find 525 nanometers spectra, you know that the Fyn is active in that place. How? Like the Fyn is inactive because basically it's not working, and Fyn will get active only when it's like doing some work. It's going on and like phosphorylating some other kinase or stuff. So when the kinase is active, it sort of gets an open conformation. The kinase detaches and SH2 and SH3 domain is exposed. And then you see there is this binder, red binder, which goes and attaches to the SH3, and which brings M-cerulin and M-venous close to one another. Like, so our basic criteria for Fred to happen was the fluorescent protein should be 60 Armstrong apart. So when this binder is attaching to SH3, that criteria is met, and basically when you illuminate it at 473 nanometer, what you see is an emission at 525 nanometer. So this was the biosensor which was made in our lab. And on the right side, obviously you again see an inactive kinase or an active kinase. So I guess I have talked a lot about Fyn kinase. Do you need me to explain the C part also?
I have a question.
Yeah.
So you mentioned that this is sort of developed as an inspiration from the last one.
Yeah.
So are there any drawbacks in the last one which is sort of removed by this new censoring method?
A very good question. Yeah, there are a lot of drawbacks. So, do you guys want me to talk about all the drawbacks and how conceptually they jump to the other like designs? They optimized into other designs. Obviously, you do design optimization when you find some shortfalls or some problems happening inside in one sensor, right?
Yes, of course.
So the problem here was, do you see a Fyn substrate attached to this Y? Like in this picture, you can see the slide, right?
Yeah.
Huh. So in the picture on the right side, so you see a fret happening. So previously this guy, they found Fushi technique, which is basically just fluorescent protein tagging and molecules moving around. Here they developed fret technique. Okay, so perfect. The technique used was perfect, but the design in this biosensor was very critically, what do you say, flawed. Yeah, you can say flawed, but still it's a design, so you have to consider something. So you see a SH2 domain exposed here.
Yeah. Which one exactly?
So you see the picture on the right side. You can see, right?
Yeah, got it. High fret.
Yeah.
Yeah, right.
Yeah, the high fret picture where the SH2 domain is like khali, ekdam se. It's doing nothing. So
Yeah.
that is the place that gets phosphorylated. Okay. And
Okay.
your fret goes out. So that is what you see in the low fret picture, right?
Okay.
Yeah, I'm asking you understood that, right? That
Yeah. Got that.
So in the high fret situation, the SH3 domain is khali. Okay. So the Fyn substrate and the linker is being wobbly, and then somehow they are getting attached. Like they're somehow pulling the Y pet near to the ECF pet. So like sort of they can interact.
Okay.
But as soon as this SH2 gets phosphorylated, it pulls them apart. So your fret is gone. Okay, cool.
Okay. Yes. Fine.
Yeah. So the question, the problem here is the SH2 domain can be phosphorylated or dephosphorylated anytime, irrespective of whether the Fyn is acting or not. So yeah, obviously when the as I show that it will get dephosphorylated when it's active. On the picture on the left side shows you that when it gets dephosphorylated, SH2 and SH3 is open. You can see, right?
Right. Okay.
So the dephosphorylation of SH2 shows it's active. But
okay.
Problem is the phosphorylation is SH2 can happen with any phosphate.
Okay.
Irrespective of your whether your Fyn is active or not, it needs to be phosphorylated or not.
So this doesn't exactly give you whether the Fyn is active or not, in a certain sense that.
Yeah, exactly. So
okay.
that is why I said this is a very hodgepodge design, because for for sure you cannot say that this is being phosphorylated at the time like there is this certain phosphate coming and like removing the phosphatase coming and removing the phosphate and making the Fyn active. There is a random phosphate which can come and activate your tyrosine SH2 domain anytime, whether you want it or not. So it sort of gives you a false positive data.
Okay, I see.
Does that make sense? Yeah.
Okay. So in the next one, how do you improve from this in the next one?
Okay, yeah. So in this design, basically, so this design, you see, it's not dependent on phosphorylation. Like, yeah, obviously the Fyn kinase opening and closing is dependent on phosphorylation, but the biosensor binding and showing fret is not like related to the phosphorylation.
Sorry.
So this sensor needs to be phosphorylated to not show fret, right? And
Okay,
this
yes, yes.
sensor does not need to be phosphorylated to show fret or not show fret, right?
Okay, okay, okay.
Is that clear? So in the in B you can see. Hello. Huh.
Yeah, yeah.
So you still need phosphorylation or dephosphorylation for inactivity or activity of the Fyn kinase, but while designing the biosensor, you do not need it as a factor. You know, like so there the phosphatase was getting eliminated by some random phosphatase. Like it will give you false data, but here the binder is specifically binding to SH3 when it's active, right?
Okay, okay.
So there is no way that you get a false data, as in it can't bind to the SH3. In the picture B, the binder cannot bind to the SH3 when it's closed.
Okay, okay. So basically the difference being that SH2 can be phosphorylated at any time, but over here you're doing, you're phosphorylating, you're binding with SH3.
Yeah, exactly.
I see.
So in that sensor, you were playing with SH2. So it wasn't established, right? With which domain you can bind and sort of make. So they're doing all kind of possible things they can do. So like design optimization is happening still now. So this was the design they made, and this was specific. The binder was specifically made for SH3 so that they can show fret. So as we move on, you'll see that this picture also has a lot of fuss in it. Sorry, a lot of what happened? What is this? Anyway, then you can see this picture has a lot of shortfall to it. We'll move on to it later. So we go next. Then we come to a biosensor, which is conformational biosensor. So in the start, I told you guys that there are two kinds of biosensor. One was substrate based and one was circularly permuted biosensor. Sorry, conformation based. So it's a bit confusing. I have to go back to the original CPGFP barrel structure to more talk about this. So first of all, I'll go back and introduce you to the beta barrel structure again. So this was the beta barrel structure of GFP on the A. In the A picture, you all can see. So there are certain parts which was in the start of alpha helix. You can see that there is this alpha helix chain that is protruding out at the top of the barrel, where you see alpha helix written, right? Hello.
Yeah.
Guys, I need response. So alpha helix. So the part which is demarcated as alpha helix was cut from a particular position. Okay. And
okay.
Alpha helix from the bottom, where you can see alpha helix polypeptide structure is written. From that part, so it was sort of cut from here and cut from here, okay, and they were sort of like you put this string and connected up, and you put that string. So when you cut a protein, you get a N and a C terminus, N termini, N terminus and a C terminus, right? So when you cut it from the up alpha of the alpha helix, you get two ends, N termini and a C termini, and when you cut it from down of this alpha helix, you get N terminus and a C terminus. So what these guys did was to take the N termini from the niche wala part and connect it with the C termini of the upar wala part, and took the N terminus of upar wala part and connected with the C. Sorry, yeah, I said N, right? So they took N from the upar wala part and connected with the C niche wala part. So it's pretty confusing. So what they did, you can see in the picture A.
That is not a continuous chain or something. That is not a continuous chain or something.
Yeah, it is. It's a beta barrel structure. It's continuous.
No, could you go back there once?
Yeah.
So, so from the top of the alpha helix, where alpha helix is written, so there are multiple, let's say there are multiple N terminuses over there.
No, like if you cut, so it's connected, right? You see it's connected. So
Yes.
you can see the intact structure, right? Here, the beta barrel structure.
Yes.
Okay. So when you cut it from a place, let's say from any of the alpha helix place, you cut it. Okay. So,
Okay.
so the cut end will be one will be N terminus and the other will be C terminus, right?
So one will be the part of the beta barrel structure that's already there, and one is the cleft part that you have just removed.
No, I didn't remove anything. I just, okay. So imagine a sheet of paper, okay, or imagine a number line, okay. So imagine a number line which goes up to N, maybe, okay.
Okay.
And then you take N plus 1, okay. So there is this position N plus 1 जहाँ पे तुम्हारा एक protein रहेगा, right? Hello. And there is a protein at the place N.
Yes.
So you cut at the place where N and N plus 1 is there. Okay. तो तुम्हारे पास क्या रहेगा? You get two different sheets with which is like from 1 to N maybe. And then you get another strip which is N plus 1 and the further numbers which were there, like further proteins which were there, right?
That's what I'm saying. When you're cutting, you get two different pieces. One is the part that is from the beta barrel structure.
Exactly.
And one is the removed part.
Yeah, exactly.
Okay.
So now imagine that you're taking the N plus 1 strip, the strip which is starting from N plus 1, okay. And let's designate that is the N part of the protein, okay.
Sorry?
The N plus 1 of the strip which you cut out, is the N of the new strip you get, right?
Okay, yeah.
And the end of that strip is C.
So the N plus 1 part is the C part or the other part?
No, so okay, मैंने कुछ गलती कर दी. So you take a number line which is say infinite maybe, or how do you say it? Maybe infinite. So you cut it at a place N, okay, and you get two strips, and the second strip has N plus 1 at the start, right?
Okay, go ahead.
So अब तुम वो N plus 1 वाला जो strip है, वो लेके you connect it with the first strip which has numbering from 1 to N.
So you connect the N plus 1 part to basically to 1.
Yeah, exactly. Now you get the point.
Okay.
So this beta barrel structure is sort of a continuous structure which is not evident with this. So like if you get this beta barrel structure, then you sort of get, like people who does protein, like they might get it, that you connect one N terminus with the C terminus of the other part, and they you basically get a new N and a new C terminus, right? So
Yeah, yeah.
the N1 strip, which was like at the end, which then becomes the starting point of the number line or whatever, or the strip.
So in a certain way, you're basically continuing the strip, isn't it?
Yeah, sure. Just you're changing positions.
All right.
Like, so now in this slide, you see A, right? The A picture. Okay.
Yeah.
So here they have numbered it, right? So there is this EGFP starting from 1 to 144. Okay. And there is this calmodulin which is starting from 2 to 148. Calmodulin. Okay.
Okay.
Hello.
Yeah, fine.
And there you see EGFP, which is 149 to 238, and which is on the left side connected to M13. Okay, so right now you do not need to focus on M13 and calmodulin. You just focus on EGFP part, right? So here it is numbered that from 144. So there is one strip which is cut. So EGFP को तुम्हारा एक बीटा बैरल है, ठीक है? EGFP का. So you cut it at the point of 144. Okay. Then उसके आगे का जो EGFP है, which is shown here, which has 149 to 238. So this is basically protein positions numbered, right? एक beta barrel structure में there will be many proteins which makes up the beta barrel structure, right?
Okay.
So you sort of अगर ये simple EGFP strip होता तो क्या होता, 1 से लेके it would have gone up to 238. But what we do is we cut it at 144, and then जो 149 वाला position है, maybe they have cut in the middle थोड़ा और, and जो 149 वाला position है, they have permuted and again. Okay, so